All right, let's go ahead and continue chapter 8. So, chapter 8 is on genetics. And we've talked about how DNA synthesis works. We've talked about transcription, translation. And we've talked about operons. So we've talked a lot about different facets of genetics. Now we can talk about what happens when there are changes to the gene. So any change to the sequence of DNA is called a mutation. Oftentimes when we think about mutations, we think of them being bad. Or if you grew up in the 80s and 90s like I did, you think of them as how you get Ninja Turtles. But that is not actually what we don't get Ninja Turtles. It's very disappointing when you're 8 or 9 and figure out that that actually is not what happens. When we have mutations, of course, yes, the effects can be harmful. But sometimes they're neutral. There is no downstream effect of the mutation. And every once in a while, they can be beneficial. So it really depends on the mutation, where it occurs, what the specific mutation is, whether or not we're going to see it be neutral, harmful, or beneficial. Sometimes, throughout evolutionary history, you can get a mutation that reverts back. Because remember, we're just talking about a change in a base. So let's say we had a C change to a T, replication, 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 replication. Eventually that T might go back to a C again, or G, or whatever I said the first time. So it can go back. It just really depends. Evolution is random. Sometimes students will ask me, well, why does an organism do a thing? There is no why. It evolved that way. Why did it evolve that way? Because every time a cell replicates, it acquires mutations. There's no why. There just is. It's mutations. And the accumulation of mutations and how those change proteins and how that changes phenotypes is how we eventually, get things like speciation, novel traits, different phenotypes, all through changes to the DNA. There are two broad things that can happen to the DNA that we call a mutation. One is a base substitution. A C to a T, or a G to an A, or a G to a C, whatever. We've changed one nucleotide to another. We have the same number of nucleotides, The reading frames are all the same. We've just swapped out one letter for another. Often more serious are insertions or deletions of nucleotides. We call them broadly, we wrap them up in the term indels, I-N-D-E-L, indel. A deletion or an insertion of one or more bases. And we'll talk more about why I said something like that being really damaging in just a couple minutes. So with a base substitution, one base is substituted for another. This frequently occurs during replication. Nothing is perfect. DNA polymerase is pretty good. On the order of, if we have 3 billion base pairs in our DNA, generally every time our cell replicates, there are 3 mutations. 3 out of 3 billion, 1 out of a billion, that's pretty good. No matter how much Grammarly you use, you will never type an essay that perfect, where you have only 1 in a billion errors. So that's pretty good, okay? But think of our cells. Every time a cell replicates, there's 3 errors. Then it replicates again, now there's 6. Then it replicates again, and 9, and 12, and 15, and so on. Okay. So that's what's happening with a common base substitution. So sometimes that happens. You get the base substitution, and because of that degeneracy of the amino acid code, the idea that you have a number of codons that might give you the same amino acid, we can sometimes experience a substitution, so an actual change to the DNA that gives us the same protein, the exact same amino acids. We call that silent. It is a silent mutation because the change in the base in DNA is not reflected in the protein. The protein looks the same. Sometimes, like the more scientific way they'll talk about it, they'll call it a synonymous mutation. You get the same, right? A synonym, you get the same amino acid. Okay. Silent is totally fine for this class. So if you have a mutation and you get the same amino acid, that is a silent mutation. And that's not terribly interesting. What's more interesting? More interesting would be what we call a missense mutation. With the missense mutation, what we're talking about is now that change to the DNA, swapping out the G for the T or whatever, has actually led to a different amino acid. And so that's what they're showing you here. In the top panel, our sequence of DNA gives us methionine, lysine, phenylalanine, and glycine. Perfect, that's great, wonderful. Very short protein. But now we've swapped out the C, indicated by the red arrow up here, for a T, which means our RNA, instead of having a G, now has an A. And now we have methionine, lysine, phenylalanine, and serine. Is that a big deal? I don't know. It really depends. What protein is this? Is this in the active site, right where it's supposed to bind its substrate? That might be a problem. Is this where you were forming a disulfide bridge to help with the three-dimensional structure? That might be a problem. Is it just kind of somewhere off in a corner? Maybe it's not a problem. Missense doesn't always mean it's a problem. It just means that now you have a different amino acid. Where do we almost always have a problem? Well, we almost always have a problem when our base substitution results in what is called a nonsense mutation. This is when swapping out one base gives us a stop codon instead of an amino acid. So in this example, we're looking a little bit closer to the beginning, and we have swapped out a thymine for an adenine. So now we have, instead of an A here in the mRNA, we have a U. So our protein is now methionine stop. That's probably a problem. You no longer have a full-length protein. The earlier in the protein this occurs, likely the more damaging. If it's like the last amino acid, maybe not a problem. But not the last amino acid, probably a problem. So it's just kind of that random luck of what type of base substitution did you get and how does it affect the downstream protein. So of the base substitutions, usually a nonsense mutation is likely to be the most harmful. But again, I don't mean to say that missense mutations are not also harmful. A disease like sickle cell anemia is caused by a single base substitution. It's a single base that swaps out a single amino acid in hemoglobin. It's a really big deal. So sometimes swapping out one amino acid can be a very big deal. But a nonsense mutation is where we end up with a stop codon. Now, where do we often have even more problems? With our indels. Our indels give us what we call a frame shift mutation. So a frame shift mutation actually changes the reading frame itself that shows up to the ribosome. And so when we're looking at the example that they've given us here, they have, it looks like, okay, so here in the original we had AAA in a row. Now we have just AA. We've removed the third A. So our original protein was methionine, lysine, phenylalanine, glycine, stop. Our new protein is methionine, lysine, leucine, alanine, who knows? Because we've changed the way that we've read the protein. So that could go on who knows how long. It could result in a very early stop codon. You know, we don't know. But we've completely changed the rest of the protein. So I have a graduate student working in my lab, and she has one strain that behaves really, really well, one strain that is defective for a lot of virulence phenotypes in bacteria. So we sequence the genome to see, like, well, what's the problem here? It has one nonsense mutation that could be playing a role, but we don't think so. The other one is a frame shift. It's missing one base that introduced a premature stop codon in one of the proteins of the electron transport chain. So that's probably what makes it defective for growth. So we see an early stop. The message gets garbled and it's no longer the same thing, right? You know, if I write something like the big red cat, that makes sense. If I have a base substitution that gives me a missense, the big red rat, well, it's a different message. I can still read it, and for the most part, it's mostly the same. But now I get rid of these two, and I have to read it like this. The no longer makes sense. Though when I think about taking the GRE again, that actually is kind of what my brain does. Nope, not going to do it. Okay, so it doesn't make any sense. Or if I oh, and this was originally C, right? Okay. B, C, A, T. Right. Just that one insertion also garbles the whole message. So often frame shift mutations, especially the earlier in the protein they occur, are going to really alter that protein and basically make it non-functional. So frame shift mutations, anything that causes an insertion or deletion of bases is usually a problem. Now, what if we had my same example here? And I knocked out 3 bases. Because I said, 1 or 2, you can really see how that would shift. But what if I knock out 3? Is that a problem? I don't know. Is this message different from this one? Yes. What if I insert 3? Overall, like, it's sort of normal, but I still have now that weird one in the middle that doesn't make any sense. Is that going to disrupt? Hard to say. If this is the active site of the enzyme, absolutely. I've just changed the entire active site. If this is where it forms interactions, yes, I've changed those interactions. If this is something like methionine that forms disulfide bridges, yeah, maybe I've changed the whole shape of the protein. So it's really easy for us to see like how the insertion or deletion of 1 or 2 nucleotides really messes everything up. But 3 or groups of 3 can also cause big problems for the protein. So questions about what the mutations end up doing for the protein or to the protein. Yes?
How did you say it affects the ribosome?
Oh, it affects how the ribosome reads it. So it changes the reading frame. So the ribosome, especially if you have only an insertion or deletion of 1 or 2, right? It reads 1, 2, 3, 4, 5, 6. It doesn't realize that like 7 is gone. So it will read 1, 2, 3, 4, 5, 6, 8, 9, 10, and it thinks that that's normal instead of reading 7, 8, 9. So it changes the way that the protein is going to be translated. So again, how do these mutations occur? So we know, kind of basically what happens when you have a mutation. It'll probably affect the protein. So it can be silent, yay, but it could be missense, it could be nonsense, it could be a big issue with frame shift. So how often does this occur? And I told you this earlier, our replication systems and bacterial replication systems are actually really, really good. Out of every 1 billion bases, you'll have a mutation rate of 1, so 1 in a billion. Meaning every time, if you scale down how big a genome is to how many genes are actually in that genome, every time you replicate 1 million genes, 1 gene will have acquired a mutation. Especially when you're talking about multicellular eukaryotic organisms that have very complex genomes, the majority of our genome does not encode for proteins. So the chance of a mutation hitting an important gene is low. It could be in an intergenic region. It could be in an intron. The chance of it taking out a gene is pretty low. Now, in microorganisms, they don't have as much, they don't have as much DNA. They also don't have as much intervening DNA or the junk DNA. So when they get a mutation, it's far more likely that they will get a mutation in a gene. How important that gene is, it depends. But the reason it's so low is because DNA polymerase is a very good proofreading enzyme. It makes more than 1 in a billion mistakes. It does make more. But it recognizes that it has made a mistake, and it can, like if you're typing, right, you can backspace and fix your mistake. It can do the same thing. But sometimes one just slips by. And, knowing all of this, even thinking about what mutations look like in humans. We worry a lot about mutations because things like mutations to the DNA can lead to cancer, like as an acquired disease that you can get throughout your lifetime. But if every time we replicate our cell, there's only 3 mutations on average, like, why do we see things like cancer and other types of disorders that are caused by mutations happen much more frequently than we might expect? Well, the reason for that is because we are constantly exposed to mutagens. And again, not the fun kind that will turn you into a superhero, the kind that will make your DNA go bad. So a mutagen is something that causes a mutation. And it increases the rate of mutations. Normally we're talking about 1 in maybe 1 million genes. They can increase the rate to 1 in maybe 1,000 genes. You have 25,000 genes as a human. So if you are exposed to a mutagen that increases the rate to 1 in 1,000, you have 25,000 genes. That means every exposure could cause a mutation in 25 genes. So we can have these exposures that dramatically increase the rate of mutation acquisition. And so what I'm going to show you on the next few slides are some different things that can cause mutations. I'm not going to ask you, how does this chemical specifically cause a mutation, right? I'm just showing you things that cause mutations and what types of mutations they can cause. But I'm not going to expect you to remember nitrous acid does this. So nitrous acid. So we can be exposed to various chemicals that act as mutagens. They can change our DNA. Sometimes those changes, how do we get those changes, right? When we talk about DNA replication, DNA polymerase is bringing in a nucleotide, so I can see how we would get a mutation. It brings the wrong one. Whoops, my bad. But if the DNA is already made, how do we get a mutation? So often what we look at is the base becomes chemically modified in some way. The existing base is changed by a chemical, which at that point may not be a problem. But when that DNA goes to be replicated, the changed base is read incorrectly. DNA polymerase didn't do anything wrong. Replication didn't do anything wrong. It took what it saw and did the best it could. But this is just an example. When exposed to nitrous acid, which is a byproduct of burning fossil fuels, it makes adenine look a lot like guanine. So when DNA goes to replicate, right, if it's supposed to be adenine, DNA polymerase should bring in thymine. But this chemical modification makes adenine look like guanine, so DNA polymerase brings in cytosine instead. So again, I'm not going to expect you to remember exactly what does nitrous acid do, but if we change the chemical nature of the base itself, when we go to do replication again, DNA polymerase might read that base as something else because it has been chemically modified. We also have chemicals that can do what's called intercalation. They physically get into the DNA. You can see this as an example here, which can also cause, this can cause, for example, frame shifts because it causes DNA polymerase to think that there are gaps and to try to fill those gaps. So we can either chemically modify a base, we can basically trick DNA polymerase into thinking that there are gaps. And of course, really commonly, we talk about things like radiation as causing mutations. I don't know if any of you watch The Simpsons. He works at the nuclear power plant, so nuclear radiation and stuff, and then at the base of the power plant is this pond where three-eyed fish. So that also is not what happens with exposure to radiation. You don't get cool things like a three-eyed fish. Instead, you get your DNA that'll fall apart. So one really common type of radiation that we're all exposed to is UV radiation. And I think we talked about this before, but UV radiation causes what are called thymine dimers. If you have two thymines on the same strand that are right next to each other, instead of binding across the way to two adenines, they form a covalent bond with each other. And that basically, again, confuses the DNA polymerase. It doesn't know what to do. It doesn't read them as two T's. It might read them as a single T. It might skip over the whole thing. It just doesn't know exactly what to do. So that's UV radiation. But of course, there are a lot of other kinds of radiation, like ionizing radiation, so gamma rays, X-rays. These actually break the DNA backbone. And when that happens, sometimes the cell doesn't repair it correctly. So you can repair things quickly, you can repair things correctly, you don't usually repair them quickly and correctly. So when the cell wants to repair something quickly, it just starts kind of gluing things back together best it can. So you can end up with, again, a lot of deletions when you start to do that. So radiation can also cause damage to DNA. And unfortunately, we can't avoid it. You can't avoid UV radiation. You can't really avoid ionizing radiation. You can try, but if you go to the dentist, you're out of luck. break a bone, you're out of luck. And chemical exposure, I mean, those are just a couple of examples of chemicals. Basically every chemical ever, even water, can cause breaks to your DNA, which can cause mutations, which is why, this building contains chemicals known to the state of California to cause cancer. Everything causes cancer. The roasting process for coffee beans makes them carcinogenic. Frying potatoes to make potato chips makes them carcinogenic. What are you supposed to do? You can't just sit and drink water. Carcinogenic. Right? Everyday exposure to everything is what increases that mutation rate. And that's why we often see things like cancer as diseases of people who are older, because over time, right, they've had, of course, those spontaneous mutations. Their cells have been replicating longer. But they've also had much longer exposure to other types of things, various chemicals, various types of radiation. I think I told you my dad was a landscaper for a really long time. And now he's really good about wearing long sleeves and wearing pants and wearing a hat. I mean, but, out there in his short sleeve t-shirt and his shorts, no hat, he's got to have the skin cancers removed all the time. Did he need them removed when he was 40? No, but now that he's in his 70s, yeah. So that's why we typically see these types of diseases affect older patients. But what can our body do to fix these things? Can it do anything? Yeah, every once in a while. So humans have evolved a mechanism to repair something like, for example, a thymine dimer. We can repair some of these things. If we couldn't, the first time you got a really bad sunburn, you'd have given yourself skin cancer. And I think you've probably all had a really bad sunburn. And hopefully none of you have had skin cancer. But we have a mechanism that basically recognizes that there's a thymine dimer as best it can, right? Not perfect. Tries to cut those out and then allow that gap to be filled. We call it nucleotide excision repair because it's cutting out the nucleotides and trying to repair that area. We know that it works pretty well because we know that there are patients who are defective for this mechanism. There's an inherited condition called xeroderma pigmentosum. And if you Google pictures of patients, you can see how awful their skin looks. They constantly have lesions. They often die very early of things like skin cancer. They cannot go outside into the sun. People will call it a sun allergy. They are not allergic to the sun. The sun is literally killing them. The UV light kills them. Okay. Most other organisms, though, have a very effective mechanism. Humans lost it during our evolutionary history. But there are enzymes called Photolyases that are capable of using light energy to drive the repair process. But we just don't have those. Bacteria do. So that's how they can, how does a bacterium that is constantly exposed to the sun in the soil not get completely fried by thymine dimers and die? Well, because it has a very effective mechanism to repair thymine dimers. They still get them, they can just repair them. So then we use that nucleotide excision repair. Okay, questions about what can cause mutations and how we fix them?
Is cancer more genetic or is it more caused by environmental?
It depends. There are some conditions that the cancer can be inherited in terms of, I'm sure you've heard of like the BRCA1, like for breast cancer. And what happens is you inherit a mutation in a gene already, and then throughout your lifetime you get a mutation in the second copy, and that's what makes you more prone to things like breast cancer. It's a defect in the cell cycle. So those of you who are frantically studying for your cell molecular exam, maybe you talk about that kind of stuff in that class. And if you don't, Dr. Bissonette offers a class in cancer biology that's, my students are in, they say it's very, very interesting. But yeah, so it can be inherited, but it also can be, some people, oh, cancer runs in my family, that's usually what they're talking about. But other people, I don't know anybody that has cancer. I don't know where it came from. That's just spontaneous.
And some people can repair better than others?
Yeah, some people have more efficient repair mechanisms than others. Some people might have, a condition that causes defective repair. Certain medications can predispose people to cancer. So like people who take anti-inflammatories, like steroidal anti-inflammatories for things like rheumatoid arthritis are far more prone to like leukemia because of just the way that the drugs work. So yeah, a lot of different things can cause those increases. But overall, what's happened is there's something gone wrong with the DNA repair, something gone wrong with the immune system, and that can lead to cancer, whether it's inherited or spontaneous. But again, that's why we don't often see cancer in children, because they haven't had a chance, right, to acquire all of these mutations through their lifetime. But sometimes we do because of very, very bad luck. Any other questions about mutations and their effects on the cell? So how does the state of California know that everything causes cancer? Are they assuming? Probably, but no, we know. So how do we know? Well, and why are we talking about this in microbiology? Because we can use bacteria to determine whether or not a chemical has the potential to be carcinogenic. Because what we look for is an enhanced mutation rate. So the idea here is that any chemical that can cause an enhanced mutation rate in a bacterium would very likely be able to cause an increased mutation rate in something like a human, which would then make it a potential carcinogen. So that's kind of the thought process here. If a chemical causes an increased mutation rate, which we can really easily observe in bacteria, and plus nobody gets mad when you mutate bacteria, but if you start, zapping mice, PETA will knock on your door. But nobody cares about the bacteria. So we do what's called an Ames test. So an Ames test allows us to expose bacteria to a chemical and determine whether or not there is an increased rate of DNA mutation. And we do this using one of my favorite bacteria, Salmonella. So what we have as scientists is there is a strain of bacteria that has been mutated in such a way that it can no longer produce histidine. So they call it histidine minus. So it has a mutation that prevents histidine synthesis. So if you want to grow it, you have to grow it on media that has extra histidine. So what scientists will do is set up an experiment where you have a control tube. So no additional chemical, you just have your culture of Salmonella. And they add, so they do work with mice a little bit, sorry. You add rat liver extract. The reason for that is because a lot of chemicals are processed by the body in the liver. And so if the chemical has to be modified by the body in order to be mutagenic, if you add the liver enzymes, you can see that modification. Sometimes chemicals don't do anything in the body if they're not modified by the liver, or they're not modified by, an enzyme, and they can pass directly through the body. But if it gets into the body, it's likely it could be processed by the liver, so we want to see that potential. So our experimental sample is going to be our potential carcinogen, our chemical, with the liver enzyme in the Salmonella. Okay, so remember, this Salmonella cannot grow without histidine. So what we do is we take our control and we take our test and we plate them on plates that lack histidine. Remember, the normal bacteria should not grow on a plate without histidine. It doesn't have the ability to synthesize histidine. What we expect is no growth. No growth, because they cannot use histidine. Or they cannot make histidine. They cannot make histidine, and we haven't given them any. You need it, you don't get to have it. You can't grow. But remember, there is a level of spontaneous mutation in every system. So what we look for is on the control plate, do we get any spontaneous mutants to form? So on the control plate, there are 2 colonies. So that means as all of the cells in the tube were replicating, a couple of them acquired a mutation that allowed them to make histidine again. So now they can grow. So you always have to have a control. And then we look at our experimental plate. In the cells exposed to the chemical, do we get the same or more than what's on the control plate? And what we see there is more. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So here, only 2 cells acquired the mutation, so just through spontaneous replication. Here, 14 did. So just kind of eyeballing it, it looks like whatever this chemical is caused an increase in the mutation rate over the spontaneous rate. So if I were doing, you have to repeat it multiple times and do your statistics and blah, 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 blah. But if I were just looking at these results, I would probably say that that chemical is a mutagen. It increased the mutation rate. And if it is a mutagen, it therefore could be a carcinogen. This chemical is known to the state of California to potentially cause cancer. Does it definitively cause cancer? No. Do I need to test that? No. But I know it causes an increase in DNA mutations, so it could cause cancer. But the other thing to highlight here is why bacteria are so interesting for studying mutations. They replicate so quickly and in such large numbers. Billions and billions of cells in that little tube overnight, and only 2 of them acquired a beneficial mutation at the spontaneous mutation rate. So sometimes people will talk about, well, I don't believe in evolution because I can't see it. I'm like, well, okay. Look at a culture of bacteria and you can. But if you think about humans on the scale of bacteria, in 1 milliliter of that, there are probably more salmonella than people have ever lived on the planet, first of all. So let's say we got to a point where there had at some point been 1 trillion human beings. They also replicate every 20, 30 minutes. We replicate every 25, 30, 40 years, depending. And then we stop. We don't do that again. Most of the time you replicate 2 to 4 times, maybe more if you're ambitious. 2 once was enough, but you get the BOGO, it's all right. Okay. I didn't want to be outnumbered. Bacteria get outnumbered, right? So they replicate that quickly. So for multicellular eukaryotes to see that accumulation of mutations that might finally give us a new phenotype that could be beneficial is just a scale we can't imagine. But luckily we have these bacteria that allow us to do these types of tests where we can see that after, the 20 generations that occurred in that tube overnight, in that population of trillions of bacteria, we got 2 that reverted. So we can see that type of evolution happen. We can see the acquisition of these new phenotypes, just like in the video of antibiotic resistance, right? They acquired resistance to 1,000 times the amount of antibiotic that should have killed them in 11 days. And I can't do much in 11 days, let's be real. But they got whole new phenotypes in 11 days. So using bacteria to do these types of tests, even though bacteria don't get cancer, and a mutation in bacteria is very different from a mutation in humans, gives us a lot of insight into our human biology. So I'm going to go ahead. We can skip slide 73. It's just about the plating process. We'll skip to slide 78, so you don't have to know anything about the plating process. Okay. So in your notes, like we skipped 73 through 77, we went directly to 78. Pass go, collect $200. Okay. So questions about mutations before we get into this kind of, I think this is like the final section in this chapter on how microorganisms share genetic material. Okay. So how do microorganisms share genetic material? We know they do. And of course, we talked about this idea of vertical gene transfer. Boring, right? You pass your genes from one generation to the next. Everybody does it. Every species does it. Not that interesting. Species reproduce. Awesome. Some of them clone themselves. Cool. Some of them don't. That's okay too. What's really interesting and unique among the prokaryotes is this idea of horizontal gene transfer. That members of the same generation can acquire genetic material from one another, which gives them a new phenotype as they are living. And I think I gave the example before of one of my daughters has blue eyes. There is no way that she could give me, all of the genes that gave her blue eyes. I can't be like, oh, girl, those eyes are cool. Can we share? No, that's not what happens. I had my chance when I was born. Before I was born. I had my chance and I blew it. Got my brown eyes. I can't exchange genetic material. Hey, you're really tall. I would like to be tall. You have beautiful curly hair. I would love to have beautiful curly hair. Whatever it is, we can't do it, but bacteria can. And they do it through 3 major mechanisms: transformation, conjugation, and transduction. And don't get mad at me about the names. I did not come up with the names. They all start to flow together, so I apologize. Transformation. So what transformation is the ability for a bacterium to pick up DNA from its environment and incorporate it into its own genome. So it literally is like, oh, look, a piece of DNA. Incorporates it into its own genome and can then theoretically make whatever protein or whatever output comes from that gene. So there was a really classic example in the 1920s that started to give us an idea that bacteria could do this. So this is an overview of the experiment, but let's break it down piece by piece. So this was an experiment conducted by Robert Griffith in the 1920s. And he knew that if you inject a bacterium that we now call Streptococcus pneumoniae, if you take Streptococcus pneumoniae that has a capsule on the outside and you inject it into a mouse, the mouse will die. When you try to isolate bacteria back from the mouse, you get bacteria that have a capsule. So you inject them with encapsulated Streptococcus pneumoniae, they die, you pull out encapsulated Streptococcus pneumoniae. We call them smooth, the smooth bacteria, because they have a capsule. Okay, excellent. Well, he also knew that there were some strains of Streptococcus pneumoniae that did not produce a capsule. Those are called rough. And when he injected mice with the rough bacteria, not terribly surprising, they lived. He was able to pull out a few rough bacteria from the bloodstream of the mice. But what we know of the capsule is that, well, we know now, right? It can prevent clearance by the immune system. It prevents things like phagocytosis. It helps them to adhere better to the tissues. So without that capsule, it kind of makes sense. Well, the immune system can recognize them. They don't adhere as well. So the mouse was able to fight off the infection. I don't know that it remained healthy. It still could have gotten sick, but it didn't die. Then he thought, okay, because the question is it the bacteria that are killing the mice? Or is it the capsule itself that kills the mice? Like, is the capsule toxic? So he killed smooth bacteria. But they have a capsule, but they're smooth, but they're dead. So he puts them in the mouse and the mouse lives, which, in the 2020s, if you put a dead bacteria in somebody, they're not going to get sick. But they didn't know. So you had to test. So the capsule itself isn't what kills the mouse. The capsule is not toxic. It's not pathogenic in a dead bacterium. Okay. Now let's take two things that we know don't kill a mouse. We know living rough bacteria don't kill a mouse. We know dead, smooth bacteria don't kill a mouse. If we put them together, what happens? So when he put them together and injected the mouse with that cocktail of living rough but dead smooth, the mice all died, which was unexpected. Two things that don't kill you, you put them together, now they kill you? How did that happen? And when he pulled the bacteria out of the dead mouse, he recovered only smooth bacteria that, of course, were living. So there was another set of experiments after that that determined it was actually DNA, because there were some, hypotheses. Can the living rough bacteria basically pull the capsule off the dead ones and put it on themselves? What is happening? And so they eventually figured out that those dead smooth bacteria, as they, kind of fell apart, released their DNA into the environment. The living rough bacteria can pick up that DNA, incorporate it into their genome, and start to produce a capsule. But it's all random, right? What is the chance that you, as the individual rough streptococcus, are going to grab the one piece of the smooth DNA and it's going to make you a capsule? The chances are very, very low. But again, when you have a population in the millions or billions, it becomes far more likely. And you only need one. Now I make a capsule, so I start copying myself, and all my descendants make a capsule, and all their descendants make a capsule, and then we kill the mouse. Now this idea of this ability happening in nature, we do know it does happen. Microbes can do this, but not very many. This is not a terribly common way that bacteria can acquire new genes during their own lifetime. Bacteria that are capable of doing this, of picking up extracellular DNA and incorporating it into their own genomes, are called competent. And there are very few competent bacteria, but Streptococcus pneumoniae happens to be one of them. So again, just by random chance, if you pick up that piece of DNA that allows you to do a cool new function, you have the cool new function. And so it's this idea of, this sounds wild, come on. Out of a 4 million base pair genome, you picked up the one little piece, because they don't pick up the whole genome. You picked up the one little piece that gave you a capsule, but when you talk about the scale of millions or billions of organisms doing this, that chance gets a lot higher. What's the chance that I win the Powerball? Pretty low. But if I got everyone in California to buy a Powerball ticket, what's the chance we get a winning ticket? But I want to split the money with you guys, sorry. But your chances go up the more people that do it. The more bacteria that are out there grabbing pieces of DNA, the chances go up that we pick up a cool one. So that's why you have to think about these things that sound like this is just such a random chance occurrence. It is, but when you have the numbers behind it, that random chance occurrence almost becomes a guarantee. If everybody in the US bought a Powerball ticket, would somebody be a winner? Almost certainly. But again, are they going to want to split it with everybody else? You all get $2. Right? No. That's why we like those things that have bad odds. Okay.
Some bacteria are capable of taking up fragments of DNA from their surroundings and integrating the fragments into their own chromosomes by recombination. Cells capable of taking up DNA from their environment are said to be competent. Only a few genera of bacteria are naturally competent, such as the Streptococcus pneumoniae in this example, but other bacteria, including E. coli, can be made competent through simple laboratory treatments. The process of transformation was first observed by Frederick Griffith in 1928 while developing a vaccine for Streptococcus pneumoniae. Griffith observed two strains of the species, an R strain and an S strain. The unencapsulated cells of the rough or R strain of Streptococcus pneumoniae did not cause disease. When mice are injected with these cells, their immune system efficiently deals with this invader and the mice live. Cells of the smooth or S strain of Streptococcus pneumoniae produce a capsule that allows them to escape phagocytosis by the body's defensive white blood cells. When mice are injected with these cells, the mice die.
I feel like it's really dramatic. I like it.
Heat can be used to kill the smooth strain of Streptococcus pneumoniae. When mice are injected with these heat-killed cells, the mice live. Surprisingly, when mice are injected with a mixture of harmless heat-killed smooth strain cells and harmless live rough strain cells of Streptococcus pneumoniae, the mice die. Live smooth strain cells can be found in the blood of these dead mice. The harmless rough strain cells must have gained the genes to produce capsules like those of the virulent smooth strain cells.
Okay, so again, it sounds like a simple experiment, and it kind of in many ways is a very simple experiment, but it really allowed scientists to, one, identify this really cool phenomenon in bacteria, but this is also one of the foundational experiments that led scientists to finally determine that DNA was the genetic material. Because there was a debate for a very long time about what the genetic material was. DNA didn't really make sense because it's such a simple molecule. There's only 4 bases. For a long time, people thought it was proteins because proteins are far more complex. There are more types of them. They're made of more complex building blocks. How could you possibly make something as complicated as a human using something as simple as DNA? So these types of experiments that, again, seem simple now, really were foundational to our understanding of course, microbiology, but genetics as a whole. All right, so what's another way that microorganisms can exchange DNA? This one is less of an exchange. The transformation is not really an exchange either. The dead one gets nothing out of the deal. The dead one's already dead. So it's not really an exchange, it's the living ones coming and they're little scavengers. Conjugation is similar in that one partner in this process gains something. The other one doesn't die though, so that's good. It just stays how it is. So this is a one-way transfer of DNA. It's called conjugation. Usually the type of DNA that's being moved is a plasmid, one of those extra chromosomal circular pieces of DNA. And the exchange is mediated through a structure that is formally called a conjugation pilus, but people will call it a sex pilus because that's kind of the bacterial equivalent of sex. You exchange genetic material through a structure. So how does it work in E. coli? Well, you'll have some bacteria that are called F positive. So F means fertility factor. So F positive cells have the ability, because they carry a plasmid, they can form the conjugation pilus. Cells that cannot are called F minus, and they will be the recipient. So what happens in this situation is you have the F plus cell that they're showing here on the left contains this hot pink plasmid that's the F factor, the fertility factor. So the way that this works, it shows them being kind of distant, but really the way that this works is the plasmid allows the bacterium to make the conjugation pilus. So it extends the conjugation pilus, it grabs the cell, it pulls them together, and then there's kind of this gap between the two cells while they are touching each other that allows the plasmid to flow from cell 1 to cell 2. So the way that works is that the plasmid will replicate one strand instead of both, and that strand, as it's being replicated, will pass into the new cell. And then it will make the second copy, right? They'll both have a copy now, and now both of these cells could go out and grab somebody else. But the F plus cell gets nothing. It is not a two-way exchange. And so again, it's one of those where people will ask, well, why? Why would a cell give something without getting something in return? And not because we can't believe that cells are selfish, but where's the benefit here? You have to build the pilus, you have to replicate the DNA. It's kind of intensive. So evolutionarily, why does this happen? Does this make sense evolutionarily? Well, think about it this way. You are in a situation where, I don't know, something is happening in your neighborhood and you're the only one that knows what's going on. Are you just going to sit at home and be like, well, I hope it doesn't affect me? Maybe you talk to your neighbor and you're like, hey, I have this piece of knowledge. You're not giving me anything in return. I'm going to share this knowledge with you. I'm going to share this knowledge with you. And they share it with and now you have a whole neighborhood together that knows what's going on. So when whatever bad thing happens, it's very unlikely to take you out. So think about bacteria. If you have one bacteria that is resistant to antibiotics in an infection, everybody else will die because the antibiotics will kill them. And then the immune system is like, hey, and the whole population is gone. But what if, well, if you're antibiotic resistant and I'm antibiotic resistant, now we're less likely to be like totally wiped out. But if we're all antibiotic resistant, the population as a whole survives. So it's not that they're trying to be generous and save their neighbors. It's totally self-preservation. That's why animals, like when they have their babies, don't just like leave the baby out. Like, oh, there's my baby. And the lions can come up and eat it, right? They protect. They gather around because it protects their population. Even if it's not their own baby, right? You're less likely to get eaten if there's a whole bunch of you. You're less likely to get picked up by the immune system if there's a whole bunch of you. So it allows for the sharing of traits that protect the population as a whole, which means each individual has a much greater chance of survival. So it's not because they're altruistic, they're bacteria, they don't think. It's because of survival. Every once in a while, bacteria can take that plasmid and actually integrate it into their chromosome. And that just keeps it more stable. It allows the retention of whatever's on that plasmid basically forever. Because the challenge with a plasmid and I think we've talked about this before, is that when the cell replicates, if that plasmid is not absolutely essential in that given moment, it is entirely possible that that plasmid goes away. Like, right, the more things that you try to carry just like independently, how likely are you to leave something behind? But if I put, you know, everything back in my bag, I'm far less likely to leave something behind. So when a cell can incorporate genes from a plasmid into the chromosome, every time that cell replicates, all of its descendants will get it, but also that cell itself is not going to have lost the plasmid. So it will always have that phenotype, whatever it is, usually antibiotic resistance. So we try to keep what's important to us as close as possible. I mean, my water bottle is not that important, I don't care. But yeah, my computer, for example. Okay, so that's why sometimes cells might integrate the plasmid into the chromosome. They don't always do it, but sometimes they do. Okay, when you have a cell that has integrated it into the chromosome, what will sometimes happen is that this cell will make a copy of part of its chromosome and give it to another cell, but that part of the chromosome may not contain the factor that allows them to do genetic exchange. So that's kind of the trade-off here. This cell, right, is capable of sharing some of its DNA with another cell, but it may not be the DNA that allows that trait to spread through the population. It might just be a random part of the chromosome. But me and all my descendants are going to keep it. So even if we can't give it to anybody else, now at least me and all of my descendants will always have it. So there's the trade-off of maybe not being able to spread it throughout the current population once you've integrated it into your chromosome, but you've really enhanced the ability to do that vertical gene transfer. So sometimes they integrate it, sometimes they don't.
E. coli cells containing an F factor, also termed the fertility factor or F plasmid, are capable of forming proteinaceous rod-like structures called conjugation pili or sex pili. Cells containing an F factor are called F plus cells and serve as DNA donors during conjugation. Cells lacking the F plasmid are F minus cells and serve as DNA recipients during conjugation. At the start of conjugation, a conjugation pilus extends from an F plus cell to an F minus cell and pulls the cells together. The cells come together and stabilize, probably through fusion of their cell membranes. The transfer of DNA begins. A single-stranded copy of the F factor is transferred from the F plus cell to the F minus cell. Each cell synthesizes a complementary strand of the plasmid, resulting in a complete copy of the plasmid in each cell. After conjugation, both cells contain a copy of the F factor. Therefore, both cells are now F plus cells and can go on to initiate conjugation with other F minus cells.
Conjugation. Okay. So what we'll do now is we'll pause here, and you can work with your small group, and I want you to draw an operon. Doesn't matter if it's inducible or repressible, but draw and explain an operon. And when you all are done, you're free to go suffer in cell molecular. Yay! You could tell Dr.